Greetings everyone, today we are going to discuss about centriole. What are centrioles? These are minute, non-membranous organelles. These are minute, non-membranous organelles present in animal cells only. These are absent in plant cells. These are present in animal cells only. And these are minute, non-membranous organelles present in animal cells only, not present in plant cells. And these are having hollow-like structure, hollow structure. They are having hollow structure, apart from which these are having hollow shaped structure, apart from which this hollow shaped structure is arranged in such a manner that first and foremost it is a hollow shaped structure, and secondly when it comes to the structure, this basically helps in uh, formation of spindle fibers during cell division, apart from which these structures also centrioles are basically involved. These are involved in the formation of vessel body, which we are going to further discuss. What is vessel body, how is it important. These are involved in the formation of vessel body, as it consists of microtubule. They have a microtubule structure, apart from which, as they are having microtubule structure, their structure is in such a manner that they are having 9 triplets of microtubules. They are having 9 triplets of microtubules on the peripheral side, 9 triplets of microtubule at periphery, while in the middle there are no triplets. There's only a protein which is present in the middle, which is known as the central hub. In middle there is a protein which is on a central hub, and on the side by side there is 9 triplets of microtubule at the periphery. Now in this case, this type of arrangement in which there are 9 triplets of microtubule at periphery and a protein central hub in between is known as 9 plus 0 arrangement. 9 plus 0 because 9 represents the 9 triplets of microtubules on the periphery. 0 represents the central uh, area in which the microtubules are present. Since no microtubule is present in the center, this is 0. 9 plus 0 arrangement. Apart from which there are 27 microtubules collectively. And when it comes to one microtubule, this is only the number for one microtubule. When one microtubule is arranged perpendicular to another one, it is known as a centrosome or diplosome. Diplosome Y, diplosome di represents two, diplo represents two, some represents body, which means two central body, two bodies which are present to the center of the cell, which are present near to the nucleus, which are present near to the center of the cell, which are present near to the center of the cell, near to the nucleus, which are present near to the center of the cell, near to the nucleus. Why? Because of the formation of spindle fibers during cell division. Apart from which, these are organelles which are devoid of DNA for self-replication. These are organelles which can self-replicate, and these are devoid of DNA for self-replication or duplication. Apart from which, we are having a fact that here these are present one in number. Centriole is present one in number. Then during S phase, they duplicate themselves. During S phase, this is a complete centrosome. The area from where two centrioles are arranged perpendicular to each other is known as centrosome or diplosome. And these centrioles which are arranged perpendicular to each other during S phase duplicate themselves to produce another centriole, a pair of centriole to produce another pair of centriole. This complete pair of centriole during S phase duplicates to form another pair of centriole, due to which both pairs of centriole are arranged near to the nucleus, and during cell division they again move to the opposite poles, due to which they undergo process of forming spindle fibers, and then assembling them, and generating force towards the chromosomes to attach the system chromatids, and again disassembling occurs. In that case, this is the process in which that occurs. Now these are involved in controlling the spindle fibers. I mean, they go to the opposite poles and produce a spindle fiber, assemble and disassemble it. Next, there is these are the centriole pair as they produce opposite structures, and they produce their structure in the opposite side. And we are having another thing I would like to discuss here, that here these are having, now these microtubules are arranged in such a manner that here we are having the microtubules. We are having the microtubules over which this microtubule, as you can see on the screen, there the microtubules are having names: A microtubule, B microtubule, C microtubule, A microtubule, B microtubule, C microtubule, and so on. In every chain, the C microtubule is connected. The C microtubule originates a bridge towards the A microtubule, which is known as the CA linker, CA connective, CA linker or CA connective. After which there are spokes which are coming towards the central hub or central road, which is known as the radial spoke, which are 9 in number. Apart from which there are spokes coming toward the center, they are known as a central spoke or central spoke or central, these are known as central spoke uh, or radial spoke. These are actually the radial spoke which are connected to the central road or central hub. Apart from which, these are triplet fibrils. We can also call them as fibrils. Why? Because these are made up of fibrous protein. Microtubules are made up of fibrous protein, so these can also be called as microfibrils. There's no doubt in it. Apart from which, a central road, central hub is a term quite clear that there is a protein to which the 9 microtubules at the periphery are attached to it. And then there is mesial satellite, pericentriolar satellite, which controls the movement, which gives the direction to centriole to create spindle fibers, as well as gives direction to centriole to either duplicate itself and give spindle fibers. That's all about this video. Hopefully we meet again.